Hi, my name is Erin Riggs, and I'm a genetic counselor and assistant professor at the Geisinger Autism and Developmental Medicine Institute. I'm also the co-chair of the ClinGen Gene Curation Working Group and oversee a number of different gene curation expert panels in the neurodevelopmental clinical domain. Today I'm going to be talking to you about the role that gene disease validity assessments can play in variant classification, as well as walking you through an example of how you might assess variants in novel genes. First, let's briefly review our case example. As a reminder, this is one of the cases you worked through yesterday with the Seeker team. Our proband is a 10-year-old female with a history of left hydronephrosis identified on prenatal ultrasound at 20 weeks gestation. Subsequent postnatal ultrasound revealed a left multicystic dysplastic kidney and a right duplicated collecting system. There was no evidence of reflux or obstruction. Genome sequencing on the proband, her parents, and an unaffected sibling revealed a de novo Misense variant in BIC1. Before thinking about how to classify this specific Misense variant, we first need to get a sense of what is known about BIC1. Has it ever been associated with disease? If so, what exactly has been observed? A quick check of OMIM shows that it does have a phenotype associated with it, but notice the brackets around the name. This indicates that at this time, OMIM is reporting this as a susceptibility to a multifactorial disorder, in this case a disorder they have termed renal dysplasia cystic susceptibility 2. Checking in ClinGen, which does indeed have several expert panels focused on kidney disorders, reveals no current curations. To clarify, a result like this simply means that the gene has not been evaluated at this time. It does not necessarily mean that the gene is not a valid cause of disease. Let's look broader now at the Gene Curation Coalition, or GenCC. In case you're not familiar with this resource, this is essentially a ClinVar for gene disease validity classifications. Here you can see the aggregated results of many other research and clinical efforts performing a gene curation assessment, including ClinGen and many others. In our case, there has been a curation of this gene by Ambry Genetics, a clinical testing laboratory in the United States. They evaluated this gene for the condition described by OMIM, that renal dysplasia cystic susceptibility 2, in 2018 and determined that there was limited supporting evidence. You'll see on the right side of the screenshot that there is the ability to click to see if there's any additional information, but in this case there isn't. We know what their conclusion was and when they did it, but we don't have access to the information they assessed and why they came to that conclusion. Finally, when we check in OrphaNet, a database of rare disorders based out of Europe, we see that they have asserted that this gene is associated with autosomal dominant polycystic kidney disease, but we don't have any further information. We don't know when they determined this, what evidence they looked at, how strong it was, etc. So why does it matter how strong the gene-disease relationship is? Recent guidance from the American College of Medical Genetics and Genomics, or ACMG, recommends that genes of uncertain significance should generally be excluded from disease-focused multi-gene panels. When encountering variants identified in these genes with little evidence supporting their relationship to disease on exome or genome sequencing, ACMG recommends additional requirements for reporting, stating that the disease relationship has not been established and separating them from any other potentially clinically relevant results to the extent possible. The reason for this is that if there's not sufficient evidence demonstrating a gene's role in a particular disease, it's not possible to accurately classify variants within that gene. It's kind of like a circular argument. If we can't say for certain that variation in general in a particular gene causes a disease, how could we ever assess any one variant in that gene for its role in disease? The 2015 ACMG AMP sequence variant interpretation guidelines explicitly state that they are not intended to be used to evaluate variants in genes of uncertain significance, and that variants in these genes should always be classified as variants of uncertain significance. When there's little supportive evidence for the gene-disease relationship, we can't assess the types of evidence we need in order to make a variant classification. Later guidance from ACMG suggests other limits on variant classification based on gene disease validity. In addition to the prior recommendation that variants in genes with limited evidence not be classified above VUS, they also suggest that genes with moderate evidence should rarely exceed a classification of likely pathogenic, given that understanding of the gene-disease relationship is still evolving. Overall, the strength of evidence supporting a gene-disease relationship has direct implications for the classification of variants within that gene. ClinGen has developed a semi-quantitative scoring metric to assess this evidence and provide an overall gene-disease validity classification. We reviewed how this scoring system works in more detail in our previous workshop, and if you'd like more background information, I encourage you to review that video and/or the slides from that presentation, which can be found at the link below or by scanning the QR code on the screen. Briefly, the classifications take into consideration both evidence for and against a gene-disease relationship. For gene disease pairs with evidence supporting the relationship, the classifications range from definitive to limited. Definitive gene disease pairs are those with considerable supporting evidence, so genetic evidence with or without experimental evidence, that have been replicated and upheld over time, in general at least 3 years. If a gene disease pair has considerable evidence available but has been newly discovered, so within the last 3 years, it will be classified as strong. Once more time has passed and no contradictory evidence has emerged, then the classification can be moved up to definitive. Moderate and limited gene disease classifications have less compelling evidence, respectively. The category of no reported evidence is reserved for those genes that may be suspected to be associated with a particular disease. So, for example, they may belong to a gene family known to be associated with the disease, but in which no human variants have been reported. If there's evidence available to contradict the gene-disease relationship, the pair may be classified as disputed or refuted, depending upon the strength of the contradictory evidence as compared to the supporting evidence. Clinical validity classifications are subject to change over time as new evidence emerges, and as such, classifications should always be dated and periodically re-reviewed. So now, let's go back to our case and the variant in BIC1. Looking through what is currently documented about this gene, there did not appear to be a clear consensus on the level of evidence supporting this gene. OMIM has it listed as a susceptibility gene for cystic renal dysplasia, and the way Orphanet has it listed suggests that it's a bona fide gene for autosomal dominant polycystic kidney disease. And at least one clinical laboratory submitted an assessment to GenCC saying that the evidence was limited. So who is correct in this situation? Often, in situations like this where there doesn't appear to be a clear consensus and there is evidence to suggest that it was last evaluated some time ago, remember that date on the GenCC evaluation was 2018, it may be prudent to do a new review to see if new information has emerged to clarify some of our questions. With that, I'm going to walk you through an evaluation of the BIC1 gene using the ClinGen gene disease validity framework. So let's start with some basic background information about BIC1. It's located on chromosome 10q21.1 and encodes a homologue of the RNA binding protein bicaudal C that was initially identified in Drosophila. It contains 21 exons and spans at least 72 kb. It contains an N-terminal RNA binding domain consisting of 5 Kh domains, as well as a C-terminal SAM domain. The SAM domain is non-essential for mRNA binding, but it is required for the transfer of BIC1 targeted mRNAs to Peabody-associated AGO proteins for silencing. Overall, the gene product BIC1 is thought to be an RNA binding molecule that functions to regulate diverse proteins at the post-transcriptional level. Within NOMAD, the gene is not particularly constrained for missense variation, and its PLI score is below the 0.9 threshold historically used to suggest that a gene was intolerant of loss of function variation. However, the LUF score, or the loss of function observed over expected upper bound fraction, that orange box there, is 0.34. This indicates that only about 34% of the expected loss of function variants were observed, and that the gene therefore is likely under selection against these types of variants. Now, normally I would review the case level genetic evidence supporting this gene-disease relationship first, but in this case I think it makes the most sense to start with the experimental data. As you'll see across the next few slides, we knew some information about the function of this gene before we ever identified any cases in humans, and in fact, observations of variation in the animal models led to the identification of the first human cases. Here I'm showing you the experimental evidence scoring metric in the ClinGen gene disease validity framework. As you can see, we consider things like the gene's underlying function, evidence that normal function is affected in a way that makes sense in the context of the phenotype if the gene's altered, model organisms, and rescue experiments. Each category has a suggested number of default points as well as a range. So if a particular piece of evidence is compelling, we may choose to upgrade, or if a piece of evidence is less than ideal, we can choose to downgrade. Each category also has a maximum score so that more convincing, more direct pieces of evidence, such as model systems and rescue, can contribute more to the overall evaluation than other pieces of evidence that might be less specific or more abstract. So, for example, it may not always be clear how changes at the cellular level relate to the overall human phenotype. In total, experimental evidence can receive up to 6 points. With that, let's look at the available animal models. So in the mid-90s, 2 spontaneously occurring mouse models of polycystic kidney disease were described. The first, listed on this slide, BPK, is a truly spontaneously occurring variant in an inbred mouse strain. Homozygous animals presented with cystic dilatation of the renal collecting ducts, biliary dysgenesis, and death by 4 weeks of age. The second variant was originally induced after exposure to chlorambucil in a different strain of mouse. These homozygous animals present with numerous cysts in all parts of the nephron, large dilatations of the intrahepatic, biliary, and pancreatic ductules, and death by 10 days. Of note, some heterozygous JCPK mice also develop cystic renal disease as they age. Over time, mapping assays suggested that these mice had variants in the same gene. In 2002, Cogswell et al. Showed that both BPK and JCPK mice had variants in BIC1. BPK has a frameshift variant in exon 22 near the SAM domain, and JCPK has a splicing variant in exon 3 in the KH1 domain. Later, targeted BIC1 knockout models were generated. The first, in 2009, involved a deletion of exons 5 through 11, and these homozygotes displayed heterotaxia of the visceral organs as well as renal and pancreatic cysts. The second, involving a deletion of part of exon 4 and all of exons 5 through 8, resulted in reduced vitality among homozygotes, with the surviving animals demonstrating a similar phenotype of polycystic formations and defects in left-right asymmetry. As in the spontaneous models, at least one publication suggested that heterozygous mice also developed glucose intolerance, diabetes, and cysts with age. Since we have multiple independently derived mouse models with consistent phenotypes, we can award the maximum 4 points in the animal model category. I'll also note that similar phenotypes have also been observed in Drosophila, Xenopus, and zebrafish, but because we've maxed out this category, I will not review this information here. Next, let's look at expression of this gene. So evidence from both mouse and human studies suggests that there is strong BIC1 expression in our organ of interest, which is the kidney. Of course, just because a protein is expressed in our organ of interest does not necessarily mean that aberration of that protein causes our disorder of interest, but this is certainly interesting information that fits within our emerging picture. For this, I will award the default score for expression evidence, which is 0.5 points. Evidence also suggests that BIC1 interacts with other proteins known to cause cystic kidney disorders, specifically PKD1 and PKD2, which are both associated with autosomal dominant polycystic kidney disease, and HNF1B, which is known to cause the renal cyst and diabetes syndrome, or RCAD. In renal tubules, HNF1B and PKD1 stimulate BIC1 expression. BIC1 in turn promotes PKD2 mRNA translation by inhibiting MIR17. Here I'm awarding a 0.5 points for each of these interactions for a total of 1.5 points. At this point, we've maximized our experimental evidence, so we've got the full 6 points. There is other evidence available in the literature, but I think we have what we need to make the case that there is compelling gene-level experimental evidence. So next, let's move on to the genetic evidence. Here, I'm showing you the scoring metric for case-level data, so variant date evidence plus segregation evidence, as well as case control evidence. Note that, just as with the experimental data, each type of evidence has a suggested number of default points as well as a maximum score. For the variant evidence, each variant type has a suggested default as well as some specific suggested upgrades. So for example, if the variant is de novo and or if it has supportive functional data. The maximum number of points that genetic evidence can contribute to the overall evaluation is 12. I mentioned before that the original patients were identified after much of the experimental data we just discussed, and in fact, they were identified because of what we learned from that information. In 2011, Krause et al. Decided to screen a cohort of 137 individuals with renal abnormalities with or without early onset diabetes for variants in BIC1 because the phenotype observed in some of the mouse models seemed to overlap that of RCAD. Previous screening of HNF1B in these individuals was negative, but no other genes related to renal abnormalities or diabetes were assessed. 2 variants in BIC1 were identified. The first variant is a nonsense variant, absent from NOMAD, identified in an individual with a history of unilateral dysplasia with cysts of the right kidney, identified on prenatal ultrasound at 22 weeks gestation. Postnatal ultrasound again showed right renal dysplasia with cysts, along with increased size and hypoechogenicity. There were no abnormalities of the liver or pancreas, and the child was said to have normal growth and development at 18 months of age. The variant was found to be inherited from the patient's unaffected father. The author did note that this father was assessed via renal ultrasound, fasting blood glucose, and hemoglobin A1C, all within normal limits. The authors went on to do some functional characterization of this variant. So previous experiments had demonstrated that BIC1 is an inhibitor of the WNT beta-catenin signaling pathway. On the right, the authors are showing that wild-type BIC1 is able to inhibit beta-catenin signaling by 60%, but the truncated BIC1 was unable to do so, with levels remaining the same as the control, which was an empty vector, suggesting complete loss of function. Remember, the default score for putative loss of function variants is 1.5, with the potential to upgrade for compelling functional evidence. While the presenting evidence is compelling, there are also some questionable aspects about this case, namely the fact that the variant is inherited from an unaffected parent and that other possible genetic causes have not been ruled out. I've opted to keep this variant at its default score of 1.5 points. The second variant is a Misense variant observed in an individual with a history of dysplastic left kidney, originally identified on prenatal ultrasound in the 2nd trimester. Postnatal ultrasound again showed a dysplastic left kidney as well as a hyperechogenic right kidney. By the age of 9 months, it was determined that the left kidney was non-functional and compensatory hyperfunction of the right kidney was observed. By age 3 years, the left kidney appeared as a cluster of cysts on ultrasound. No abnormalities of the pancreas or liver were observed, and at age 5 years, the proben was said to have normal growth and development. Like the previous variant, this variant was also inherited from an unaffected parent, the mother. In this case, no specific ultrasonography or lab data were provided, but the authors noted that family history was negative for renal failure or diabetes. This variant is also observed in 3 individuals in NOMAD. However, given the prevalence of polycystic kidney disease and/or congenital renal anomalies, along with the genetic heterogeneity associated with those conditions, the reobservation is not outside the realm of plausibility for a causative variant. Further, because many individuals with polycystic kidneys or congenital renal anomalies are developmentally normal, it's not inconceivable that some affected individuals could possibly be present in the NOMAD cohort, though of course the cohort is in general depleted for severe early onset disorders. Krause et al. Also performed functional assays on this variant. Again, we are looking at BIC1's ability to inhibit beta-catenin signaling. Here, the authors are showing that this missense variant results in a 22% reduction in inhibition activity compared to wild type. Of note, this variant is within the SAM domain, so the authors also demonstrated that the patient's missense variant behaves similarly to that of a complete deletion of the SAM domain, also about a 22% reduction in activity. The default score for missense variants is 0.1 points, with the option to upgrade in the presence of functional data or if the variant was de novo, etc. Again, like the last case, the functional data is interesting, but the same questionable aspects remain. The variant is inherited from an unaffected parent, and other possible genetic etiologies have not been ruled out. Here, I've opted to give this variant 0.5 points. Very recently, just this year, a third case has been reported. Note that this evidence has come out after that last 2018 date of evaluation we saw in GenCC, demonstrating why it's a good idea to do a current literature search when thinking about using an existing gene disease validity assessment. Here, Johnson et al. Reports on a cohort of 100 fetuses or stillborn cases with severe renal disease. They did targeted NGS on 155 genes involved in monogenic kidney disease and identified a single variant in BIC1. This is a de novo splice variant that is absent from NOMAD, observed in a proband with a different renal phenotype, this time bilateral renal agenesis. This individual also had a myelomeningocele, though it's unclear if this finding is related to the BIC1 variant. There is a family history of a full sibling with a fetal myelomeningocele, but no DNA was available for testing. Here, we have no functional data to confirm that this variant is indeed resulting in a truncating protein. However, we do know that this variant is de novo, so I will upgrade the putative loss of function variant score to 2 points. So let's return to our case. How would they fit in this overall evaluation? So remember, our case, it's a de novo missense variant observed in a 10-year-old female with a history of left multicystic dysplastic kidney and right duplicated collecting system. This variant is present in NOMAD in a single individual, and as we discussed before, I don't think that's enough to rule this variant out as a plausible cause of disease, given the frequency of renal anomalies in the general population. We do not have functional data in this case, but because it is de novo, I am upgrading the default missense score to 0.5 points. So overall, we have 4.5 points of genetic evidence and 6 points of experimental evidence for a total of 10.5 points. As you can see in the red box, this falls within our moderate category. Note also that the range of the moderate category is from 7 to 11 points. So we discussed earlier the possibility of, maybe should we be downgrading the 2 originally reported cases for various reasons. Remember, those were the ones that were observed in unaffected parents, and the other genetic causes hadn't been ruled out. I just want to point out that even if we had downgraded those, or even if we had discarded those completely, if you still believe the Johnson et al. Case and our case, then this gene-disease relationship likely would still come out to moderate based on the strength of the experimental evidence. That's really putting a lot of weight here. Sometimes it's helpful to think about whether or not the overall classification outcome would be changed when you're having difficulties deciding how to score an individual piece of evidence. So in summary, there is moderate evidence supporting a relationship between BIC1 and congenital anomalies of the kidney and urinary tract. If you'll recall, all of the currently proposed disease entities focus on the polycystic kidney aspect, as that was what the main phenotype of the 2 originally reported cases. With the newer report having renal agenesis, it may be appropriate to consider a broader term to describe the totality of the evidence we now have. The term congenital anomalies of the kidney and urinary tract is a generic one describing a group of abnormalities affecting the kidney and other structures of the urinary tract, and can include both unilateral and bilateral presentations of some of the specific anomalies we've observed in our cases. So hypodysplasia, renal agenesis, multicystic dysplastic kidneys, along with many others. There are both syndromic and non-syndromic presentations, and variable expressivity and reduced penetrance have been described. So this is, again, something that we also observed within our cases. Overall, it would be reasonable to report this variant back to this patient. Of course, a separate assessment would be needed to evaluate the evidence supporting the pathogenicity of this particular variant, much like what the previous speakers just reviewed. However, because of your gene disease validity assessment of moderate, you now know that most variants in this gene would be unlikely to warrant a classification higher than likely pathogenic. And with that, I will stop and take any questions. Thank you.